Okay. Good morning and welcome everyone to this workshop on single-cell applications in environmental health research. And what I thought I'll do is, because many of the speakers who are going to follow me today are going to get into specifics of various applications of single-cell and single-molecule technologies, I thought I'll try to give you an overview from a disease context, try to highlight some of the examples of single-cell applications in human health and disease, and towards the end, talk a little bit about some of the work done at the Broad Institute with Aviv Regev and Orit Rosen, who's here today, on how this might be applied to the gut. So broadly, when we think about disease, we have sort of two sort of basic factors. So I think all of us in this room and who've joined this call, this meeting will agree that there are clearly genetic factors that contribute to disease, and clearly many of the diseases have environmental implications of why somebody with a specific genetic variant might get disease, might as well as not get disease. And broadly, these applications affect disease states. So for example, in the lung, diseases such as asthma and obstructive disease, chronic pulmonary obstructive diseases, two diseases that have a huge influence on environmental pollution and smoking, effects of toxicity, whether they be environmental stressors or medications that we might take, and more broadly, how we respond to treatment. And we've sort of measured these influences for the most part by measuring events or effects in peripheral blood, with the hope that some of these might reflect what might be going on in lung in the context of obstructive pulmonary disease, in the states of toxicity in the liver, in diseases of liver toxicity, and, for example, in the context of cancer, how tumor cells respond to treatment or develop resistance. But what I like to, the first sort of take-home point I'd like to make is that this is suboptimal. The peripheral blood is a poor readout of what might be going on in organ physiology, pathology, and disease. And one might need to begin to think about what are these specific cell types that maintain homeostasis in these various organ systems, and how one might be able to measure these cell types in disease and health. And so what I thought I'll do is, in the first part of my presentation, talk about some of the applications around this concept, and then get into some specifics. So I've listed a bunch of different diseases here, from inflammatory bowel disease, a disease that affects the gut, allergies that are increasing in incidence and throughout the country, and neurodegenerative disease, cancer, and various other diseases. Again, all of them have clearly genetic contributions to the disease, and in many of these conditions, we believe that there are environmental influences that influence the disease. But for the most part, we rely on diagnosis based on histopathology, so an established state under the microscope, with paying very little attention to cell types and cell states. And what I'll try to argue is that cell types at single cell resolution might help us to identify how a specific disease is initiated, how these diseases might progress, how one might get toxicity to various treatment responses and complications and so forth. So that's disease and genetics. What do we do in trying to understand the environmental influences of in this context? Is broadly sort of we measure various factors in the environment, and these are mostly gross measurements. And then on the internal environment, this can move from various states of the microbiome to specific influences on organs. And broadly, sort of this is combined to a measurement of the exposome, so work that's been done by Steve Rappaport and Martin Smith at UCSF have clearly identified that these interactions need to be measured, and hopefully what this workshop will try to achieve is that these need to be measured at a single cell resolution. So what do we do now? Sort of broadly try to measure the impact of environmental influences by measuring the exogenous events, and then again, as in the disease state, mostly sort of a drop of blood in sort of trying to measure some of these consequences. So what I'd like to, again, revisit this concept and try to highlight how genetics might be better appreciated by understanding the single-cell states of genetics, and how environmental factors might be better appreciated by understanding the environmental influences. So 2 examples. So for example, if you take COPD, chronic obstructive pulmonary disease, there's elegant studies done by many authors that have identified a number of genetic factors that contribute to the 2 states of the disease. So clearly genetic factors that affect the large airways and a subset of genetic factors that affect the alveolar structures. And genetics has also allowed us to move on to trying to bin these genetic variants with some of the complications. So a subset of patients go on to develop pulmonary fibrosis, and I'm sure we'll hear about that later today. And then a subset of patients have overlap syndromes. And clearly this disease has a huge environmental influence in its onset and progression. A second disease that I thought I'll highlight is the emerging disease burden of steatohepatitis, or basically what we refer to as NASH. And by 2020, it's expected that the largest number of transplants for liver disease is going to be due to this underlying condition. Again, genetics has been identified, but again, if we are able to sort of map these genetic variants at a single cell resolution, we might be able to get to what might be some of the initial events that might contribute to disease. So what has been done to sort of try to dissect this contribution here, and I thought I'd take you through 2 examples. So I'm going to highlight how single cell mapping of the liver has helped us to sort of get at least a basic framework of how liver disease and toxicity to environmental influences could be measured. And then work again from Aviv Regev and J. Rajgopal from the Broad Institute on how mapping the single cell resolution of the map of the lung might influence this. So a paper that was published in 2018 went on to sort of map the epithelial compartment of the lung, which basically went on to identify many different cell types. But I think the key finding was that they were able to, for the first time, identify the cell type that now expresses the CFTR gene. So the cystic fibrosis gene is expressed in a specific cell type called the ionocyte. And prior to this single cell map, one sort of presumed that this transporter was assembled or expressed in a broader category of cell types. So obviously moving genes to a specific cell type to function. The second example is with liver disease. So if you look at the textbooks of medicine, the liver has a lobular architecture. There are zones within the liver. There is a central vein that drains many of the breakdown products centrally. And there is a sensing system or a defense system in the portal triads that basically involves 3 structures. So the hepatic artery, the hepatic vein, and the bile duct. There are a number of cell types that bring about this interaction. And for the most part, it's been viewed in textbooks of medicine that all hepatocytes are equal. And many of these cells within this category basically have features that are very similar to what might be in the circulation. What Sarah Polland and Gary Bader have gone on to show recently is that, in fact, now if you map the liver at single cell resolution, you get many cell types, so probably 20 distinct populations, and suggesting that these hepatocytes have unique features, so it's not one uniform mass of cells. And in a follow-up study from Ben Moshe and others, they were able to go on to show that that central vein, where the liver lobule is located, it has certain sets of functions, and that portal triad that I told you has most of the immune cell activity has a different set of factors. So clearly, a complex mass of cells, division of labor, single cell maps have allowed us to map at single cell resolution how the liver might function. So in those 2 examples, what I've tried to show you is that genetics has given us a starting point, but by understanding the single cell maps of these specific cell organs, one might be able to get from gene to cell type and potentially to how disease might occur, and how environmental factors might contribute to this. And for the second part, I thought I'd focus a little bit on the gut. Obviously, the GI tract that extends from the oral cavity all the way down to the rectum broadly is involved in 4 functions. They absorb nutrients for us. They play an important role in barrier function. They play an important role in immunity and metabolism. And the value of studying the GI tract, not because I'm a gastroenterologist, but it gives you a view to all the other organ systems. So there are many diseases that affect the gut, but we've now begun to appreciate that many diseases, for example, of the central nervous system, such as autism, may have its origins in the GI tract. And if you think about how the environment might influence, second to the lung, the GI tract is probably one of the largest organs where there might be a huge burden of environmental events. In the past, we've gone about studying this. I think Norbert mentioned about flow cytometry, is by collecting clinical and phenotypic data, isolating cell types based on known surface markers, so you can only recover the known cell type. You're unlikely to identify new cell types. And then trying to find ways of interactions between these cell types, relying mostly on static systems. What I think single-cell data is probably going to help us do is to get sort of a global or a holistic picture of that organ, try to understand how cell types might communicate with each other, and then try to predict what might happen in various perturbation states. So to do this, with Aviv, Regev, and Orit, we've sort of begun sort of trying to map the gut at single cell resolution. So can we identify every single cell type in the gut? And try to identify how these cell types might help maintain homeostasis, control inflammation, and resolve disease. So lots of cellular immunology have identified many cell types with many components within them that maintain homeostasis, control inflammation, and bring about resolution of disease. But up until this technology has been available, we've not been able to map each of these processes to these individual cell types. And I basically take you through a few examples here. So the first is that work from Hans Clevers and others have shown that the intestinal stem cell, which expresses LGR5, then develops or progresses through lineage steps to develop into many cell types. And what Adam Haber and Moshe Biton did was to try to see whether one can map these cells at an entire organ level. So they went about starting with mice to isolate many of the cells in the small intestine, and you can begin to see that there are many flavors to that cells that come from the small gut. And what single cell mapping has allowed us to do is now begin to identify unique markers for these cell types. So we're able to recover what is known, but obviously the value of single cell methodologies is to try to identify what is unknown. So one example, so as you go from the proximal part to the distal part of the GI tract, one ends up with different expression patterns and different cell types. And so one of the cell types that I briefly alluded to here was this tough cell. So again, if you go back to the textbooks, it's believed that there is one tough cell in the gut. And I'll give you an example as to why the tough cell might be important in helminth infections. But what single cell mapping allowed us to do was to now identify that there may be 2 flavors of tough cells. One of these tough cells had features that looked like an immune cell, so they expressed also CD45. So if you relied on surface markers, one might misclassify this cell as a potential immune cell. But what it went on to show was that CD45 is now broadly expressed in other cell types. And then a second cell type of a tough cell that looked very much like a neuronal cell. So suggesting that as these cells go through the lineage process, they have different steps in that early stages of development. The second cell type is the enteroendocrine cell. So again, in the textbooks, there is one enteroendocrine cell. So these are the cells that basically secrete hormones in the gut, help in metabolism as well as digestion. But what single cell mapping has allowed us to do is that there are now probably close to a dozen enteroendocrine cells that, again, have the ability to sense the environment and secrete many of its differential products. So that's what we've learned in health. The second question was, how does the gut then respond to infection? And in this particular experiment, this same mouse model was infected with a common bacteria, Salmonella, or an helminth. And that allowed us to basically ask what happens to those individual cells in the gut. And we were able to map this over time. And what I thought I'll do is just focus in on what happens in the gut following infection with a helminth. And what you can see here is that on day 3, you get this spike in that tough cell population that continues to increase as infection progresses. And that, in fact, what I'm not showing you here is that this was not a feature following bacterial infections. So again, getting back to potential environmental influences, there could be ways of identifying how environmental features might affect many of these cell types. What else did we learn? So the belief is that the stem cell then contributes to many cellular features. And when Adam Haber and Moshe was examining them, they noticed that the stem cell in the gut had maybe 3 different flavors. And so Hans Clevers believed that there was one stem cell that was LGR5 positive. These 3 stem cells all express LGR5, but based on its cell cycle gene expression, you can break them up into 3 stages. So the question was, what are the gene expression patterns of these 3 stem cell states? And as you can look at it here, a subset of these stem cells expressed MHC class II machinery, suggesting that this stem cell could potentially function as an antigen-presenting cell. And further inspection of the genes that were expressed in that stem cell subset that expresses antigen-presenting capability showed that, in fact, it had many other features that could do this process. And if you then follow this in a dynamic manner, you can begin to see by fluorescence microscopy in a dynamic healthy gut, you can see these T cells marked in red coming and making contact with that stem cell that expresses MHC class II, suggesting that this was an ongoing interaction. So what might be some of the implications of this? So for example, if you were to then delete MHC class II expressing stem cell, now you in fact expanded that LGR5 positive cell, suggesting that it played an important role in maintaining homeostasis. And I'm not going to go into details, but what we were able to then also show was that this interaction with the stem cell and T cell played an important role in educating the T cell responses. So in the case of the helminth infection, this interaction promoted by Th2 cells expanded the expression of goblet cells in the gut, which is what we might see. And then similarly in a bacterial infection, in the context of Th1 immunity, there was a different lineage that expanded. So what I've tried to highlight in the second part of my presentation is the value of getting into detailed mapping, mostly focusing on single cell biology, where we might begin to understand new rules of regulation of organ systems, but then also translate that in the context of a disease setting. To end, I thought we might sort of revisit the disease context. So just last week, there was a paper again from Brad Bernstein and colleagues that basically revisited a disease, acute myeloid leukemia. So this is a disease that is diagnosed based on expansion of a leukemic cell population. And it's generally viewed that these cells all have similar features. But what Brad and colleagues were able to demonstrate is that, in fact, there are 7 stages to these leukemic cells. And in fact, the treatment response depended on how many of these stages might be present or detected at single cell resolution. So what I've tried to do in the last 15 or so minutes is to give you a broad overview of how single cell methodologies might be applied to health and disease, what might be needed to try to better appreciate environmental influences in health and disease. I've on purpose stayed away from methods because I'm hoping that Orit will talk about it in this session that follows and other speakers. But then the value of sort of better defining disease and how environmental influences might bring about health and disease. And finally, obviously, the next sort of revolution in single cell methodologies is to sort of try to map these single cells at an organ level to try to identify how these cells might contribute to a disordered organ architecture and various disease states. So I'll end by saying that what I've tried to do was to sort of try to highlight the value of trying to better understand what might be going on at a single cell level in various organ systems and how this might be the beginnings of better mapping on quantifying cell environmental cell influences in health and disease. I think I'll stop there and happy to take questions.
Thank you very much for the talk. It's extremely interesting and a great framework for moving forward through our workshop. I'm curious about when we talk about, as you start to look at cell types within a given organ or tissue, you're identifying different subpopulations based on expression of different markers and patterns of that. But I wonder whether we're also seeing, even within what you would call a single cell type, continuum of different expression patterns, and how fine do we go in terms of categorize something as a different cell type versus a different variation of a cell type? I don't know. How do we look at these things in terms of how much lumping and how much splitting we want to do for the purposes of understanding mechanism and response?
Sure. I mean, I had it on one of my slides, and I should have spent a little time trying to better define cell types and cell states. And I think one of the challenges that we are likely to run into as we map various organs at cell type level resolution is changes in expression patterns might identify cell states, so the same cell having many different coats that do certain things in health and potentially do certain different functions in disease. So I think the challenge always is, as technology comes into a field, you end up splitting things. But I think as long as we stay with the goal of trying to understand how health and disease might be influenced, defining cell types should probably be the primary goal. And so again, in the stem cell example that I gave you is, again, likely a feature of cell state based on cell cycle gene expression and the expression of MHC class II in a subset of those cell states. Okay, good. Thank you. Oh, sorry.
Rajwar Gepalli from Thomas Jefferson. It's fascinating with the single cell atlas distribution that you have shown. One question is, are there any insights in terms of the evolutionary aspect of how these distributions are either constrained in particular ways in any given tissue, or have they become more complex as you go up on whichever way you think of traversing the tree? What's the insight we have so far?
Yeah, so I think it's the early stages of trying to understand what are the signals that constrain cells within an organ system. So as we've studied or applied single cell methods so far, we still rely on separating these cells from its organ structure. And so it's hard to better define signals that limit cells to a specific compartment. And so the way we've gone about this in the case of, say, the gut stem cell is to identify gene expression patterns in a dissociated state and put them back together and try to come up with principles of how cells stay within a certain compartment. So, for example, the cells in the portal triad, the specific immune cells that are found in the portal triad in the liver, are rarely found in immune cells that surround the central vein. So suggesting that there are features that keep those immune cells in those two zones. But I think the order to sort of try to better appreciate what you've just outlined, I think one needs methods of spatial restructuring or spatial transcriptomics. I just briefly sort of showed you one slide on the slide-seq method that's again being developed at the Broad Institute, where one might be able to put these things together. And that might allow us to better appreciate cell-cell interactions. Obviously, we'll hear about this later today, where with single-cell measurements, we only get a snapshot of the transcripts. We don't measure every single transcript in a cell. And I think hopefully that methodology will also get better.
So the related question, if I may, is you talked about the human liver zonation and the patterns and so on and the subtypes. There's obviously data like that exists from mouse. And so from a toxicological point of view, is studying the single cell landscape in the hepatocyte landscape, if you will, in the liver, is that giving clues of why the toxicology might be different between why we're having a difficulty of translation, for example?
Yeah, so the hope is that, I mean, obviously the paper that the two papers that I briefly alluded to, I didn't justify all the excellent work in that paper. I just picked two figures from those two papers. So I think one of the work from Gary Beda and colleagues, where they were able to show how the different zones might explain. So for example, around the central vein, again, going back to textbooks of hepatology, there are 3 zones that basically tell you about why certain zones are more likely to be perturbed by toxic signals compared to the other. And I think what that initial human atlas was able to tell us is that there are certain hepatocytes and other cell types that do a lot better P450 metabolic breakdown versus getting rid of oxygen radicals might be in a different hepatocyte. So I think what needs to be done is to now go back and sort of look at it in a model system, whether these features could be better captured.
Thank you.
Thank you, Dr. Ramnik. It was an excellent talk. Shudin Bhattacharya from Michigan State. Are there any attempts to compare some of these subtypes that are being identified to gene signatures from bulk RNA-seq of disease phenotype to identify which subtypes might already be kind of midway along the path towards, say, certain stem cell population in the liver, for instance, that are kind of primed for progression towards carcinoma, for instance? Sure.
So there's probably a lot going to be done in that vein. So we've done this recently for the gut, where one uses single cell data from a small number of cells. And then once you know the gene expression patterns of those cells, one can go back to bulk RNA-seq data and reclassify the cell types that might have contributed to that bulk signature. Obviously, it has certain limitations, but I think as we sort of capture features that define each of the cell types, I think we're going to be able to go back and deconvolute that bulk signature into which cell types may have contributed to that process.
So another question, this committee often ends up talking about inter-individual variability as a factor in environmental response and environmental susceptibility. When we look at these kinds of studies and these kinds of evaluations, how much work is being done across individuals different people with the same disease state or different things. How much information do we have about how variable some of these characteristics and patterns are across individuals as opposed to, just across the cell types within an individual?
Yeah, it's an important question, and I think those types of studies are just being done, where we are trying to ask, obviously, in our own research at the Broad, we're interested in trying to understand individuals who have the same genotype as to why individual A might end up with disease, whereas individual B might be protected from disease. I think this is going to get a lot more interesting because we've also begun to identify protective variants that protect ones from various disease states. And so trying to map, I think the first step is going to be to map genetic variants to a particular cell type. And then once you're able to do that, to sort of basically do more than transcriptomics. I think you need to find out what happens at a protein level, a metabolome level, and so forth, to try to basically better appreciate individual variation and its responses to even a simple drug like statins. Why a subset of patients develop myopathies and abnormal liver function tests, whereas the majority are able to tolerate those drugs with no side effects. And obviously, the cost of these types of applications is going to be an important factor. And I'm hoping that as costs come down, one might be able to do these population-level studies. So, I've often asked the question, what would have the Nurses Health study or the Framingham Heart study looked like if we had access to single-cell technology in the first few years of that cohort? What would have been the predictive value of that data 20, 30 years on? I would think it would have been quite spectacular. But these types of applications, I think, will only advance our understanding of how these signals are sensed and how they are integrated.
So the complexity that you're reporting with all the different cell types and kind of the individuals is kind of daunting. But on the other hand, like when I'm thinking about, like, say, the multiple kinds of intestinal stem cells now that are going on to develop and are, where do we stand about understanding what the epigenetic signatures of those are? Is that what the differences are? Then you're going to back up and say, well, why? But do we have some of that information yet?
Yeah, so that information, again, is being collected. I think obviously the challenges have been, I'm sure we'll hear about sort of epigenetic measurements at a single cell resolution. I didn't want to end up including sort of overview slides that would have taken away what many of the other speakers are going to get into later today and tomorrow. But I think those are important, measurements that need to be done. Obviously, you always run into the challenge of collecting too much data and not being able to make sense of it. But I think if you take these as a process, so for example, how is an organ maintained in health? What happens in inflammation? What happens as cellular proliferation changes? And what happens when you now get oncogenic transformation and then begin to understand the cell types, the epigenetic maps that are associated with it? I think we'll begin to better dissect what those multiple signals are and how they are integrated at a cell type level. Okay, so thank you again. And so we'll move on to a second speaker, Arit Rosen, who also comes from the Broad Institute. She's the scientific director of the Klarman Cell Observatory that has played an amazing role in generating many of these data types and many of the methods. And she's going to tell us about cell atlases as roadmaps to understanding tumors. Great.